everyone! Today I'm going to be discussing how dietary interventions targeting the gut microbiota can modulate human immune status and reduce systemic inflammation. To provide some background, one of the main factors that determines the composition of the gut microbiome is diet, and the increasingly popular Western diet has proven to be really harmful. As we're all very familiar, the Western diet causes a decrease in gut microbial diversity and abundance, leading to chronic inflammation and disease. The Western diet is characterized by lots of processed foods, along with higher levels of refined sugars, sodium, and dietary fat, and reduced dietary fiber. Though the components of a Western diet have a direct negative impact on the human body, they also affect our health indirectly through the modulation of the gut microbiome, which in turn modulates our immune system. All of these factors combined lead to an overactive immune system, causing low-grade systemic inflammation that can cause all kinds of diseases, which we can see here in this diagram. Chronic inflammation can contribute to strokes, heart attacks, heart disease, obesity, type 2 diabetes, liver disease, inflammatory bowel disease, colorectal cancer, and so much more. The World Health Organization estimates that 74% of deaths globally are caused by non-communicable diseases, such as the ones we see here. If we want to reduce the rates of chronic inflammation around the world, it's really important to address the root of the issue. Studies have found that dietary interventions could counter the negative health effects of the Western diet and restore the gut microbiome by increasing microbial diversity and abundance. Making changes to your diet is an easy way to improve your health and reduce the risk of developing inflammatory diseases later in life. Unlike medications, you don't have to go to a doctor and get a prescription, which makes dietary changes so much more accessible to people. There's also the affordability. Consuming healthier, whole foods doesn't cost any more than the processed, unhealthy elements of the Western diet, and it can actually be cheaper in a lot of cases. Medicine and even probiotic supplements are pretty expensive, especially in America, and improving our diets could reduce our medical expenses throughout our lives. Dietary interventions also involve a considerably lower risk of side effects than medicines. One of the most well-known examples of a gut microbiome-friendly diet is the Mediterranean diet, which consists of whole, unprocessed ingredients, including high volumes of vegetables, fruits, cereals, legumes, nuts, and fish. The Mediterranean diet also uses a lot of unsaturated fatty acids, such as oleic acid found in olive oil, which are known to support gut health. The diet has also been found to reduce the risk of cardiovascular events like heart attacks and strokes, and it also reduces systemic inflammation, partially through the modulation of the gut microbiota. Fiber is an essential part of our diets. The human digestive system is actually not able to digest fiber, but the microorganisms in our gut can digest it. Fiber is fermented by our gut microbes as one of their main energy sources, and in turn, they convert fiber into microbial metabolites that have a beneficial impact on our health. Some examples of high-fiber diets include the Mediterranean diet, the vegetarian diet, and rural or unindustrialized diets. Research has found that high-fiber diets are associated with improved microbial richness and increased metabolites, along with better health outcomes. However, it's inconclusive whether high fiber actually improves microbial diversity. On the other hand, we have fermented foods, another part of our diet that's known to improve the microbiome and our general health. Fermented foods usually, but not necessarily, contain live microorganisms. The consumption of fermented foods can increase the bioavailability of polyphenols, which have antimicrobial activity and can inhibit the growth of pathogens in the gut. Research has found that people who consume more fermented foods, especially yogurt, show increased gut microbial diversity along with increased metabolites present in the stool. Fermented foods also reduce the risk of cardiovascular disease, type 2 diabetes, and obesity, which we all know are diseases associated with chronic inflammation. In the study we'll be discussing today, the researchers investigated the effects of 2 gut microbiota-targeted diets on the immune system. They studied a high-fiber diet and a high-fermented food diet. Participants following the high-fiber diet were consuming more fiber that comes from plant-based sources. So as a result of this, they also ended up consuming more vegetable protein and less animal protein. Foods high in fiber include fruits, vegetables, legumes, grains, nuts, etc. Participants following the high fermented food diet consumed a higher proportion of animal protein, mainly in the form of fermented dairy products. Fermented foods include yogurt, fermented cheeses, fermented vegetables, vegetable brine drinks, kombucha, etc. Over here we have a nice graphical abstract of the article that shows us the main findings from their research. They found that participants in the high-fiber group showed an increase in microbiome function, as shown by an increase in microbial proteins and metabolites, such as enzymes and short-chain fatty acids found in stool samples. These participants also had personalized immune responses to the high-fiber dietary intervention. Their inflammatory response decreased for people who had high microbial diversity and low inflammation to begin with. But those who had low diversity and high inflammation at baseline actually experienced a further increase in inflammation. In the fermented food group, participants showed an increase in microbiome diversity and decreased inflammatory signals and immune cell activity. So we can see that both the high-fiber and high-fermented food diets had beneficial yet very different effects on the microbiota and the immune system. Let's get into the experimental design. Participants were divided into a high-fiber group and a high-fermented food group, with 18 people in each dietary intervention group. The study was conducted over the course of 17 weeks. The first 3 weeks were before the dietary intervention took place, and the researchers used this time to collect information about the participants' diet, stool, and blood at baseline. They also took measurements throughout the intervention. In this figure, you can see everything they tested for. So in the stool, they looked at 16S ASVs, metagenomics, proteomics, and short-chain fatty acids. And then in the blood, they looked at levels of inflammatory cytokines, endogenous cell signaling, and cell signaling capacity. The dietary intervention took place over the course of 10 weeks, with the first 4 weeks being a ramp phase, where they slowly introduced more high-fiber or fermented foods to their diets. Then there was a maintenance phase during the next 6 weeks, in which they were required to consume a certain amount of high-fiber or fermented foods, but they were free to choose which specific foods they consumed. Finally, we had a 4-week choice period, in which participants could choose to continue the diet they were assigned to whatever extent they'd like. These graphs on the right illustrate this very nicely. So for both groups, you can see that initially, participants consumed smaller amounts of fiber or fermented foods. Then you see the gradual increase during the ramp period, and then high levels during the maintenance phase. Then we dropped back down a little bit during the choice period, but overall, participants still chose to eat much more fiber or fermented foods than they did before the intervention. Let's do a brief overview of the methodologies used throughout the study. So all of their DNA extraction from stool samples and fermented food samples was done using the MoBio Power Soil Kit. So first, for the 16S amplicon sequencing, they used Illumina sequencing to generate over 15,000 reads per sample, and each read was about 250 nucleotides long. The 16S amplicon sequencing data was then demultiplexed using CHIME, and the amplicon sequence variants were identified using DADA2. Then, for metagenomic sequencing, they used the Nextera Flex Kit for library preparation, and once again used Illumina for DNA paired end sequencing using a NovaSeq 6000. For stool proteomics, they used mass spectrometry and then added the peptide samples to reverse phase chromatography, which was done with HPLC. Short-chain fatty acids were not measured in the lab, so instead, frozen stool samples were sent to Metabolon for measurement, and the researchers used paired Welsh t-tests to determine if the levels of short-chain fatty acids changed significantly from baseline to the end of the intervention. To measure both immune cell frequency and endogenous signaling, they washed, stained, and filtered whole blood samples and used CyTOF, or cytometry time of flight. Then for serum cytokines, they sent serum samples to Olink proteomics, where they conducted an inflammation panel assay of 92 proteins to analyze the cytokines present in the blood. Finally, signaling capacity. So again, serum samples were washed, stained, and filtered, and then they used flow cytometry. Let's start by looking at the results for the high-fiber diet. This first figure shows the number of 16S ASVs, or amplicon sequence variants, found in the stool over the course of the study. The data points stay pretty consistent, and there is no significant change here, meaning that the high-fiber diet did not cause a change in microbial diversity. However, the figure on the right shows that there was a gradual increase in the percentage of microproteins found in the stool. This indicates an increase in the overall number of microorganisms in the gut. Although we're not seeing new species, we have a higher abundance of the species that were already present, which makes sense because the increased fiber in the diet provides more fuel to sustain a larger population of gut microorganisms, and more microbes means more microproteins. This way, a high-fiber diet modulates microbiota function and allows the already present gut microbes to more effectively exert their beneficial effects on the host. This diagram goes along with the last one we just saw. So along with the increase in microproteins found in the stool, we're also seeing an increase in chymes. So chymes are enzymes used by microorganisms to digest carbohydrates. This graph shows the levels of each of these different chymes listed along the x-axis, where the white boxes are the levels at baseline, while green is the levels at the end of the intervention. With the high-fiber diet, we're seeing an increase in all of these chymes over time, and I'll get to the significance of this later on in the presentation. One thing to note, though, is that in the fermented food group, they actually found a decrease in some chymes, while none of their levels increased, indicating a major difference in the way these two diets impact the host. Here we're looking at the levels of short-chain fatty acids found in the stool with the high-fiber diet. There was a decrease from baseline in the levels of isobutyric acid and isovaleric acid, which are both branched-chain fatty acids. High levels of these compounds are associated with hypercholesterolemia. There was also a decrease in valeric acid, which is often found in high levels in people with autism. It's still unclear whether these fatty acids decrease as a direct result of the increased fiber consumption, or if it's because the participants were eating less meat and dairy products, which contain these branched-chain fatty acids. It's important to note that they actually didn't find an increase in butyrate, which is probably the short-chain fatty acid you're most familiar with. Butyrate is often elevated in people with increased microbiome diversity and functions, as many studies have previously found, but we didn't find that in this study. This could have been due to incomplete fiber fermentation by the host or incomplete absorption in the colon. Now we're moving into the data they gathered from blood tests to investigate the impact that the high-fiber diet had on immune function. There's a lot going on in this figure, so let's take it one step at a time. This is a heat map showing the change in each of these immune functions. So we're looking at immune cell frequency, cytokines, endogenous signaling, and signaling capacity. Each little row you see represents one participant in the study. The blue color represents a decrease, white means there was no change, and then orange and red mean there is an increase compared to baseline measurements for each person. Now, the participants were divided into 3 immune profile responses based on their baseline levels of inflammation, and this actually influenced how their immune system responded to the high-fiber diet. On top, we have our high inflammation group, and these participants exhibited an increase in inflammatory response after the dietary intervention, which we can see based on all of the red and orange we're seeing up here. There was a significant increase, especially in endogenous signaling and signaling capacity. Moving down, we see that the 2 groups that had low inflammation at baseline exhibit a decreased inflammatory response after the intervention, as shown by all of the blue color here. Overall, this figure shows us that each individual's immune system responds differently to a high-fiber diet, and depends largely on their initial levels of inflammation. A high-fiber diet can reduce inflammation in those who already have low inflammation, but for someone with high inflammation, it can actually make things worse. Now let's take a look at the results we found for the high fermented food diet. The consumption of fermented food resulted in an increase in microbial diversity, as shown by figures A and B, which show us the number of observed amplicon sequence variants and the Shannon Diversity Index. We see a steady increase in both of these from baseline to the end of the study. The graph on the right shows us that the number of observed ASVs is positively correlated with the number of fermented food servings consumed per day. Fermented foods often contain live and active bacterial cultures, so it makes sense that diversity would increase with increased consumption. What's interesting is that the type of fermented food actually impacted the microbial diversity. We see here that the orange circles on this graph represent vegetable brine drinks, and the trend line has a slope of 7.9 as compared to the total fermented food servings, which has a slope of 2.7. The blue circles represent yogurt, and the trend line for yogurt actually has a slope of 13.8, suggesting that yogurt is highly effective in increasing gut microbial diversity, with vegetable brine drinks following closely behind. Another point to note, there's no figures showing this, but there was no increase in microbial metabolites and proteins found in the stool, unlike what we saw with the high-fiber diet. If we look at this figure here, this is showing us the percentage of new ASVs in stool samples that were shared with fermented food over the course of the study. Pay attention to the y-axis here. We're seeing only a tiny percentage, 2 to 3, up to 6% at week 6. Just a very small percent of these new ASVs found in stool samples overlap with the ASVs found in actual fermented food. The majority of these new ASVs were distinct from those found in fermented foods. The last slide we saw, showing the increased microbial diversity, it may have led us to believe that the microbes in fermented foods were colonizing the gut, but this figure shows us that that's actually not the case. In fact, this dotted line here represents the trend we see in the high-fiber group. And again, we're seeing around 2 or 3% of those ASVs shared with fermented food, even though the high-fiber group participants weren't consuming fermented foods. So this suggests that fermented foods were somehow shifting and remodeling the gut microbiome, allowing new and previously undetected microbes to thrive in the gut. Now let's get into the impact that the fermented food dietary intervention has on immune function. The researchers tested the levels of 63 different cytokines and found that 19 of those decreased compared to the levels at baseline, showing us that fermented foods can decrease inflammation by mediating levels of inflammatory cytokines, such as IL-6. What's really interesting is that none of these 19 cytokines changed in the high-fiber group. Both the high-fiber and high-fermented food groups showed an overall decrease in the levels of inflammatory cytokines, but they affected different cytokines. And just like what we saw before with the high-fiber group, the fermented food diet also resulted in a decrease in signaling proteins released by immune cells such as B cells, monocytes, and CD8 and CD4 T cells. This figure compares the levels of immune signaling proteins at baseline to week 10 of the study. All the points to the left of this dotted line indicate a decrease, and we're seeing that most of these signaling proteins did actually decrease over the course of the intervention. There was only one signaling protein on the right of this line that actually increased. This again goes to show that fermented foods can reduce inflammation in the body. So we've gathered data about the microbiota, and we've gathered data about the host immune system. Now let's put it all together. This figure shows the significant correlations between individual microbe and host assays. The light gray color represents a p-value less than or equal to 0.05, while dark gray is p less than or equal to 0.01. Starting from the top with the most highly correlated parameters, we see that immune cell frequency is highly correlated with short-chain fatty acids. We also see that host proteins are highly correlated with Kzymes. Immune cell signaling and host proteins are both correlated with 16S ASVs, and host cytokines are highly correlated with microbe proteins. These are just the first few pairs in the list, but we can see that there's clearly a relationship between microbial factors and the host immune system. We talked about Kzymes earlier in the presentation, which were those enzymes released by our gut microbes to digest carbohydrates, and we saw that the levels of K-zymes increased after the high-fiber dietary intervention, but a few of them decreased with the fermented food diet. K-zymes are actually very much interlinked with the immune system. In this figure on the left, we see a list of diseases and functions of host proteins along the y-axis, while the x-axis shows us the number of significant correlations they found with K-zymes. The blue color to the left of the line represents a negative correlation, while the magenta to the right of the line is a positive correlation. And we see that this graph is overwhelmingly blue, indicating that in most people, K-zymes are negatively correlated with things like inflammation, immunological diseases, skeletal, muscular, and connective tissue disorders, and so much more. However, some people did have positive correlations, meaning that K-zymes were associated with disease. Moving to the other figure on this slide, this one shows the impact of K-zymes on immune signaling. At the top, we see several cytokines that simulate all of these different immune cells and cause them to release P-stats, which are phosphorylated transcription factors that begin signaling cascades. This figure shows Spearman correlations between several types of K-zymes and these P-stats, and all except for one of them are negatively correlated, showing us that K-zymes are negatively associated with immune signaling, and they could reduce the inflammatory response. Here's a brief summary of the conclusions we drew from this study. The high-fiber diet did not impact microbial diversity, but the fermented food diet resulted in an increase in diversity. The levels of microbe proteins found in the stool increased with the high-fiber diet, but did not change with the fermented food diet. There was an increase in K-zymes in the high-fiber group, while we actually saw a slight decrease in the fermented food group. Branched-chain fatty acids, which are associated with disease, decreased in the high-fiber group, but no data was collected in the fermented food group. Finally, the change in inflammatory response in the high-fiber group depended on the levels of inflammation for each person at baseline. People with low inflammation at baseline had a decrease in inflammation, while those with already high inflammation at baseline had an increase. Whereas in the fermented food group, the inflammatory response decreased regardless of baseline inflammation. From this article, we learned that increasing the amount of fiber and fermented food we consume can support the health of our gut microbiome and reduce systemic inflammation through the modulation of the microbiota. Dietary interventions can be used as adjunctive treatments for inflammatory diseases, or they could be used as a preventative measure to reduce the risk of developing them later in life. However, it's important to remember that for people with inflammatory diseases who already have high levels of inflammation, consuming more fiber could actually make their symptoms worse. For these people, consuming more fermented foods would be helpful in reducing inflammation. Overall, making changes to your diet is an easy and accessible way to improve your general health, and anyone can incorporate more gut-friendly elements into their diet to combat the negative effects of the Western diet and industrialization. The study did have a few limitations. First of all, there was no control group in the study. Instead, the researchers compared the results from the high-fiber and high-fermented food groups to each other, and they compared results at different points in time, so like from baseline to week 10 of the intervention. Having a control group would have allowed the researchers to better understand whether the microbiota changes they were seeing were actually caused by the increased fiber or fermented foods in the participants' diets. Another limitation we have is the sample size. So there were 18 people in each of these groups, and all of them were healthy individuals. Although we did have the high baseline inflammation group when we discussed the high fiber diet, these people were still in good general health. Given that this entire study was conducted to determine the impact of these gut microbiota-targeted diets on inflammation, it would have been useful to also study these dietary interventions in the context of inflammatory disease. Finally, the length of the intervention may have been too short to reveal some of the longer-term impacts of the high-fiber diet on the microbiome. The maintenance period, which was when participants sustained a high level of fiber consumption, was only 6 weeks long, and we didn't see any changes in microbial diversity. However, fiber-induced increases in microbial diversity might be a slower process, meaning that consuming more fiber could actually increase diversity, but we just needed more time for the microbiome to fully adjust to the dietary changes. For future research, investigating the impact of increased fiber and fermented food consumption together could possibly yield even better results for the microbiome health and reducing inflammation. We saw in this study that the two diets both benefited the participants' health, but their mechanisms of action were different, so it would be interesting to see how they would impact health when they're put together. Future studies should also take place over a longer period of time. Although the microbiome does respond to changes in short-term studies like these, we might see different results if we allow the microbiome more time to fully adjust to dietary changes. Consuming more fiber and fermented foods should be more of a long-term lifestyle change than like a quick 2-month diet and then dropping it and going back to eating junk food. A longer-term study showing the effects of these diets over the course of a year or more would be a much more accurate representation of how they affect your health. As I mentioned in the last slide, it would be really helpful to conduct experiments studying the effects of fiber and fermented foods on participants who have inflammatory diseases, and maybe this way we could determine how exactly dietary interventions could be used to treat or at least reduce the severity of diseases caused by systemic inflammation. Here I have some discussion questions for you guys. First, how do fermented foods increase gut microbial diversity? Are the new microbes coming from the environment, or were they already present in the gut in very small, undetectable quantities? Number 2, with enough research, do you guys think dietary interventions could eventually be tailored to treat inflammatory diseases and not just manage the symptoms? 3, why did people with higher levels of inflammation at baseline have an increase in inflammation when they consumed a high-fiber diet? 4, how would fiber and fermented foods impact our health when consumed together? For example, would fermented food-induced diversity increase even more with the increased fiber for microbes to use as fuel? Would fermented foods reverse the negative impact of fiber on those with already high inflammation? I really love this question, so feel free to get creative and really think about all the possible ways that these two elements could interact with each other and influence the microbiome and levels of inflammation. And finally, number 5. Why did fermented foods change the levels of different inflammatory cytokines than the ones impacted by fiber, with no overlap between the two groups? Thank you all so much for listening, and I look forward to hearing what you guys think about this article.